Both these guys are 50. Surprised? Well, me too at the first. But more so, curious. Why don't we all look like that? Why do we have to grow older and then our bodies deteriorate and eventually we die? Can we prevent that? Can we fix it? If we replace all the organs in our bodies, would we be younger and not die? But what is the reason for this happening in the first place to know how to fix it?
Bring it back.
A body is a group of organs, and each organ performs a specific function. Each organ is a group of small factories called cells. In order for this organ to perform its function, each cell has to perform the same function on a micro level. Take for example, when your lung takes in oxygen, a huge group of lung cells has to take this oxygen and give it to the blood cells in order to distribute it to the whole body. Same with your heart. When your heart pumps blood into the whole body, it has to contract. For this to happen, each heart cell has to contract to pump the blood into the whole body. How many of these small factories do you need to make a body? An estimate of 30 to 40 trillion cells. Each cell, being a small factory, needs a manager and workers. The manager of the cell is the DNA, the workers are called proteins. As any manager, the DNA gives a group of orders for the cell to function. Each order is called a gene, and each gene is translated into a protein, and this protein executes the function. To get a picture of how this works, consider eating this potato. This potato contains carbohydrates that will get broken down in your intestine by enzymes, which are proteins, into glucose. And this glucose will get absorbed into your blood by other proteins. And then after that, the glucose will circulate in your blood and get sensed by receptors. Those receptors are other proteins on the pancreas that will send a message to the DNA of pancreatic cells to start producing insulin, which is another protein. This insulin tells all the cells in the body to take the glucose in. Because our bodies are so complex, this DNA has 20 to 25,000 genes, which is why it's 2 meters long in each cell. If you take the DNA from all the trillions of cells in the body and stretch it, it would make a trip to the sun and back 70 times. And yet this 2 meters DNA has to fit in a 6 micrometer diameter nucleus in the cell. To fit this DNA into the nucleus, we need a packaging system. And by now you should know that this will be done by proteins. Those proteins are called histones. The DNA is wrapped around those histones in a tight manner that it fits into this small size. But wait a second, what if we need to read a gene in the middle of this heavily packaged DNA? Well, only the part of the DNA that contains this gene gets loosened first, and then the gene can be read. But if this DNA is the same in all the trillions of cells, then how come we have different types of cells? It all comes to the packaging pattern. This packaging pattern is distinctive for each cell type because it allows only the genes needed in a specific cell type to be read. That's why you don't get insulin produced from your eyes. So for that to happen, each cell has labels on the genes that they need to be read. Those labels are chemical groups on the DNA itself and the histone proteins that the DNA is wrapped around. And they change according to the cell type and the cell needs. And we call this mechanism epigenetics. Now let's imagine a scenario where a piece of this DNA gets broken. Immediately, the cell sends a team of proteins to fix this part. So it loosens the DNA in the broken part and then starts to fix it. But unfortunately, after unpackaging this part and then packaging it back, it doesn't go back exactly as it was before. Over the course of time, the cell loses its distinctive packaging pattern that lets the cell know which genes to read, and hence it loses its identity. Imagine having a brain cell or a heart cell that doesn't remember what to do anymore, which leads to the organ being less functional and you get older. Look at those twin mice. One of them looks normal and the other looks a lot older, although they are the same age. But the one that looks older was subjected to multiple DNA breaks over the course of its life. This process that leads to aging also leads to the cell losing the chemical labels on the genes. Wait a second. What if we track those changes? Would we be able to tell the actual age of a cell without knowing the age of the person it came from? Turns out that there is this chemical label that is called DNA methylation that can do just that. Based on this DNA methylation, several biological clocks were developed. What is astonishing is that these biological clocks could not only predict the age, but also could predict death. But before we get to that, a couple of interesting facts about these biological clocks. Men were found to age faster than women by 4%, and the biological clock of an embryonic stem cell was found to be close to 0. What about twins? Would they have an identical age according to their biological clock? Interestingly enough, their biological age were the same, but later in life they changed it and became different than each other, which means that this biological age is in part hereditary, but also it gets affected by your lifestyle. But here's an interesting question. What if the biological age was different than the chronological age? In other words, what if your cells are actually younger or older than you are? Well, it turns out that the difference between the biological age and your actual age is a predictor for mortality. In mice, it was even more surprising. A study that used machine learning to create a program to predict the death of a mouse was accurate with a median error of 42 days only. This machine learning program were given the data of the DNA methylation and the time of the death of different mice, and then it was taught to predict the death of other mice based on their DNA methylation data. What is even more astonishing in this study is that when the mice got a healthier diet, the program predicted a delayed death. And it was true. What? The death of mice can be delayed by merely changing the diet? Whoa! Can we delay aging? Can we delay our aging? And perhaps even delay our own death? Can we have like an actual age of 50 but a biological age of 40? Turns out we actually can. Several interventions were found to delay aging in humans. From them is fasting, calorie restrictions, and some drugs like rapamycin, metformin were found to be useful. But how does that happen? With the same mechanism that aging happens, anti-aging can happen. If aging happens, due to cells losing their distinctive packaging pattern, then those drugs and calorie restrictions maintain or help to maintain the integrity of this distinctive epigenetic pattern, which means the cell do not lose their identity in an earlier age. But that tells me something. If we know how cells age, and we can also delay this aging, can we reverse it? Sounds like we need a part 2. See you in the next episode.